Hello guys, in previous lecture we had already discussed the factors of apoptosis and route to apoptosis and the enzymes that involve in the process of apoptosis. In today's lecture we will discuss the mechanism of apoptosis. Apoptosis occur by two mechanisms. Number first, death receptor mechanism, and number second is mitochondrial or intrinsic mechanism or pathway. So what is death receptor mechanism? Death receptor mechanism or pathway. This pathway depend upon the receptor called death receptor. Death receptor, and death receptor is of two types. Number first is tumor necrosis factor 1 and number second is CD95. Remember, tumor necrosis factor 1 present on cell membrane, present on plasma membrane and act as a receptor for Fas ligand. Tumor necrosis factor 1 present on plasma membrane and act as a receptor for Fas ligand. Tumor necrosis factor 1 contain intracellular death domain. Contain intracellular death domain. Intracellular death domain. For example, this is a cell and this is a tumor necrosis factor 1. It contain intracellular death domain and act as a receptor for Fas ligand. For example, this is Fas ligand. What is Fas ligand? What is Fas ligand? Fas ligand is a membrane protein of T cell. It is a membrane protein of T cell. So, for example, if there is any injury, radiation, toxin, chemotherapeutic agent, for example, this is a cell, and this cell is injured due to any injury. For example, this is a tumor necrotic factor, contain intracellular death domain. So when there is any type of injury, Fas ligand will come. Fas ligand will come and attach to the receptor attached to the tumor necrotic receptor type 1. It will combine with the tumor necrotic factor type 1. Cell contain a lot of tumor necrotic factors. For example, this is tumor necrotic factor. When Fas ligand attached to this, the death domains of the tumor necrotic factor, they comes together. The death domains of tumor necrotic factors, they comes together and make a cluster and that cluster and make cluster and that cluster is called fed fast associated death domains. These cluster the death domains will form a cluster. So what occur first? There is injury to cell. Fast ligand fast ligand will attach to tumor necrotic factor 1. The death domains of tumor necrotic factor 1s make clusters and that cluster is called fad fast associated death domains. When these domains are formed, then what will be happen? Number second, when death domains come near to each other, certain adapter protein inside the cell, certain adapter proteins inside the cell, they bind the death domains, bind to death domains. For example, this is a cell. It contain tumor necrosis factor 1 and these are they make a cluster. Then certain adapter protein comes and bind these death domains. When adapter protein bind these death domains, they activate they activate caspase number 8 and we know that caspase number 8 is a initiator caspase. So initiator phase is done. What will be the next phase? The next phase will be executioner phase. When factor caspase 8 is activated, this caspase activate caspase number 3, 6 and 7 and we know about this. These are the executioner caspases. These are the executioner caspases. So executioner cell phase is also done. What is the function of caspases? What caspases do with cell? Caspases. What it will do with cell? Number first, they will digest cytoskeletal protein. Cytoskeletal protein of plasma membrane. Cytoskeletal protein means those protein that give the those protein that gives the structure to the plasma. Number second, caspase will digest cytoskeletal protein of cytoplasm also of cytoplasm. Number 3, caspases also activate DNase enzyme. Activate DNase enzyme and DNase enzyme damage the DNA, help in breakdown of DNA. Remember that DNase enzyme break down the DNA, break down the nucleosome of DNA. Nucleosome of DNA. We know that double strand of DNA is wrapped around the histone protein. For example, this histone protein. On this protein, the double strand of DNA is wrapped around, and this whole structure is called nucleosome. Number second, intranucleosomal breakdown. Intranucleosomal breakdown of DNA is the feature of apoptosis. Number 3, intranucleosomal breakdown is done by executioner caspases. Executioner caspases. So, up to now, what we have discussed, we discussed that, for example, this is a cell. It has tumor necrosis factor 1, and it contain intracellular death domain. When there is injury or any type of toxin, the fast ligand that present on the T cell, it will combine with tumor necrosis factor 1 receptor. Tumor necrosis factor 1 contain intracellular death domain. When it combine with this, the intracellular death domains of tumor necrosis factor 1, they make a cluster and form, and that cluster is called FAD. When FAD is formed, then another type of enzyme in the cell comes and bind to this cluster, join them. When in this protein are called adapter protein. Adapter protein. When adapter protein bind the death domains of tumor necrosis factor 1, it activate caspase 8. Then caspase 8 will activate executioner caspases, which are 3, 6, and 7. When these are activated, they will digest the cytoskeleton of the cell membrane, cytoskeleton of the cytoplasmic membrane, and they will also activate the DNase enzyme, which will undergo intranucleosomal breakdown of DNA.